what we're going to do is George is going to present his slides for in the next few minutes. And if you have any questions while during the presentation, please feel free to pop them in the chat, and I'll be keeping an eye on the chat. And George is happy to take some questions as we go along. So with this, I'm going to pass over to George. George, to you.
Thank you. Hello everyone. So thanks for joining us today. As Audrey's kindly introduced me, yes, I look after the microscope facility based in the Well building called the Well Cellular Imaging Centre. We're a core facility, so we primarily serve Denmark Hill campus, but we are open to everyone within King's as well as other universities. We occasionally get people coming to use our equipment from elsewhere. We have 8 microscope systems, so a few of those are confocal microscopes. We have a live cell epifluorescence system, a pair of super resolution systems, an upright multiphoton, and a light sheet microscope, as well as a high content imaging system. We're going to be going through a bit more detail about the confocal live cell and the super resolution systems, as well as the high content. So to kind of briefly mention a bit more about how we operate as a sort of facility, our systems are available for you to come and use. We provide training and support on each of the systems that we have. We can also provide a small amount of imaging as a service. So if it's a one-off, we can maybe do the work for you. There's often discussions needed around that. I provide consultation, so where you're planning an experiment, you can ask me, what would be compatible with the systems that we have. We provide courses and workshops. So there's a Keats page that we have. If you search for Well Cellular Imaging Centre in Keats, you'll find us. And we also provide tools for image analysis and quantitation. So we've been open about 6 years now, and many of you attending today probably have already used our systems, but if any of you are interested to come and use our facility, I've got some contact details at the end of the talk. So I'm going to talk through the first sort of system type, if you like, confocal microscopy. Many of you will be aware of what confocal imaging can provide. Primarily, it's aimed at producing fluorescent imaging, but with optical sectioning, which gives you high contrast, and that means you get more precision because you're only getting the light which is in the focal plane. So a sort of typical usage you might have for that kind of system is where is my protein, or other molecules, obviously. And an example that I'm going to share is a fairly recently published example of looking at neuronal marker for innovation in aortic tissue. So the example here is some tissue, some peripheral aortic fat body tissue, if I recall correctly. Apologies if I get this wrong, but they were looking at the presence of the innovation by virtue of this target, tyrosine hydrogenase, and looking at the difference between a obese mouse model versus wild type, and showing very clearly less innovation in the fatty bodies. So this is a really useful, a good example of showing confocal imaging, because you can see very clearly where those proteins are localized within the tissue. Tissue can be quite tricky to image with a standard fluorescent microscope because you get a lot of the light from out of focus, so you don't get a lot of contrast, and it can make it very hard to interpret precise localization signals. So this was a good example of this. Another example from one of your colleagues, Andrew Cobb, recently published some work where they were looking at localization of DNA damage, which in this example here you can see some nuclei in some U2OS cells, and they were looking at characterizing presence of the DNA damage markers, which you can see in yellow, with respect to position near tubulin. No, sorry, actin. Got my molecules wrong. The benefit, again, as I said, with confocal imaging, is that you get these bright high contrast image data. And also in some cases like this, you get very visually stunning. I had mentioned we used to run an image competition, and this was one of the winning entries. So I always like to use those images where I can. Moving on, so I mentioned to Audrey earlier, you know, for each sort of system type, I'll take a moment for questions if necessary. So I'm kind of now got to the point where I'm not going to talk anymore about confocal imaging. If anyone's got a query, feel free to say. Audrey, is there anything?
So we don't have any question on the chat yet.
All right, no worries. Okay, so talking also then about our live cell epifluorescent microscope. It's a system that can do quite a few different things. It's a sort of standard inverted microscope, but it's equipped with some sensitive fluorescence cameras, of which we have a pair with a beam splitter, so you can look at two different colors in the same instant. And it also has a standard light source for fluorescence, but then a second light source for rapid switching of wavelengths. And both of those features, the sort of dual emission and what we may call dual excitation, they're separately used typically for what we would call ratiometric imaging. So ratiometric imaging, if you're not already aware, where you take images typically of two wavelengths, be it wavelengths where you change the excitation or the emission, as I've mentioned, and you can use these for readouts, functional readouts in live cells, such as calcium imaging. So a classic example is Fura2 dye, and this dye it changes its fluorescence depending on whether calcium is present nearby. There are possibly hundreds of other similar ways to read out different functional things such as ATP or voltage. So, you know, we're not limited to these things. Epifluorescent imaging, in contrast to confocal, tends to be faster because you're reading out at the speed of a camera, and typically seen as being quite gentle because you don't need to use as much light. So cells, when you're imaging them over a period of time, can be quite sensitive to intense light flux, and that's where epifluorescent microscopes tend to be an advantage. Another example of a dye that you can use for reading out calcium is fluo4, and as an example, one of your colleagues, again, Matteo Beretta, was looking at mitochondrial calcium levels caused by the calcium release with respect to presence of a particular receptor, as seen on the screen. And in the example imaged on this system, they were taking images of the fluo4 over a period of time, and determining the ratio of the signal with respect to time point 0, and then looking at how mitochondrial calcium changes with response to the presence of a histamine. So if I recall, histamine's a good way to sort of spike ER uptake, and comparing that with the knockout of the gene of interest. So this was some work that he did a while back, and I helped him with some of the cell masking. So as I mentioned before, we don't simply provide you access to the equipment. We'll also give you input on how to take the images. Our training is usually sort of bespoke for every researcher, but also for the analysis side of things, if you've got queries on how to perform something like cell masking and then ratiometric analysis, yeah, these are things that we can provide you input on. So sort of briefly pausing, as I mentioned, yeah, if you've got any queries now about the live cell system that we have, feel free to ask.
I think we can move on, George.
Yeah, all right. We have two different super resolution microscopes in the wall. One of which we've had for a while now, the iSim. So this is a system that will, in terms of how you use it and prepare your samples, it's fairly similar to a confocal microscope. The acquisition protocol is a little different, but the sort of headline is that you can get 100 nanometer resolution within fairly reasonably easily, and it's a, as I say, it's a fairly straightforward system to use, provided you put the right kind of sample on. The other system that we have is a storm microscope, which I'll go into a bit more detail exactly how that works in subsequent slides. But that can provide 20 nanometer resolution. However, the sample preparation is quite different, in that you generally don't produce sort of typical slides. You have to have your sample in some solution and use the correct types of dyes and things like that, which, as I mentioned, we provide you with consultation. So yeah, if it's something you're looking to do, we can give you the information and guidance on how to actually get such an experiment working. Moving on to an example use case of the iSim. So I've got a bit of error between my slides there. Yeah, I was going to say it's 120 nanometers resolution on the iSim. So an example from Matt Stroud's group, Ed Batty was looking at the change in shape of nuclei within some cells post-exercise and or not exercise. So some believe ex vivo cell, some tissues, and they were looking at the difference on how exercise changes the shape amongst other factors. And the iSim provided a really good way to image these and get really decent detail on the shape of the nuclei, because it can go into tissue the same as a confocal will, but yield better resolution. So in the example shown here, you can see the difference between young and old, trained and untrained, and there's a stark difference in the trained cell, this nuclear shape from trained mice, where they have a more round phenotype, and I think this is, again, visually stunning way to demonstrate what microscopy can be all about. Yeah, that's a sort of an example of how you could use iSim. Moving on to, well, sorry, again, pause. Anyone, any questions about the iSim? As I said, I'm happy to take more questions at the end. Was there any in the chat?
No, there's nothing on the chat. Matt just shared a link to a study that's just been published. So
okay, all right. Okay, so moving on to STORM. So that stands for Stochastic Optical Reconstruction Microscopy. The comical way people like to describe STORM is that it's blinking microscopy, mostly just because sometimes the sample preparation can be fiddly. That's actually got a lot easier in the last few years due to some commercial kits that you can get. The sort of basic premise on how STORM works is that you need your fluorescent molecules, termed fluorophores, to blink one at a time, or at least, on and off in a sort of random pattern. As it turns out, while you're using a standard imaging platform in terms of optics, the size of the single emitter that you get is still going to be the same size as the normal resolution of your microscope. But what you are able to do is localize where that dot is more precisely than the resolution typically is set. So this is how the limit on resolution is broken in that sense. You can localize each emitter precisely. And the way that it works to generate an image, this video that one of the early developers of super resolution imaging, Ricardo Enrique, came up with this very apt way to demonstrate. He took a phone video of the Eiffel Tower and ran his particle detection scheme on it, and then if I repeat that a few times, you can see how each of the emissions is built up to yield a higher resolution image of the Eiffel Tower. So you can imagine, to actually obtain an image like you would for a microscope image, you've got to take many thousands of images, and the process to do that, obviously we have a system which can do that just fine, but it's just so you understand kind of the basis of how that works. A study that I was able to find on some example in cardiology was looking at the presence of ryanodine receptors clusters. So within the cells, the ryanodine protein receptor clusters form localizations with another protein, which under standard microscopy you can't really distinguish them apart. But with STORM imaging, bear in mind the scale bar here is your sort of typical resolution of a microscope. You can see actually where they form clusters within localization, but not exactly always overlapping with the other protein. So it's these kinds of things, if you're wanting to look at protein localization, STORM can be another tool to use for this. And for sub-organelle morphology, that sort of thing, you can really start to look at. And I'll mention again that there's a lot more involved with the sample preparation, which, as I said, we have experience and all the information to give to you if you're interested. But it's just a sort of preface the idea that STORM imaging is very possible, but you do need to do it correctly. So were there any questions on STORM imaging?
Not on the chat at the moment, George.
Okay. I'll move on to the sort of last system now. So I was going to talk a bit about high content imaging. We have a system which is a very automated microscope designed for imaging in multi-well plates, and it's a combined system. You can work as a confocal or as an epifluorescent, depending on what you want. It has a very handy built-in analysis software, and it makes it very simple to do routine things like masking cells and then measuring parameters about them. The biggest benefit with high content imaging is that you can build assay style microscope experiments. So if you want to find out how well, what what kind of concentrations you need to use for a transfection, or just count the efficiency, or indeed viral transductions, or dose response curves, to see what happens with regards to morphological changes, as a sort of a simple example. But the amount of different types of information you can get from that kind of experiment, with the analysis module built in, you can count spots within cells, within thousands of cells, and all kinds of subcellular patterns and things. Another feature that we have on there, which I'll highlight with an example, is the intelligent scanning options. So it allows you to do an experiment where you pre-scan at a lower magnification, and then use an analysis routine to pick out cells, and then the microscope can come back and image those locations, which allows you to pick up rarer events. In the example that I'm going to give, there was two populations of cells, and it was Dipali Patel, who was in Kinya Otsu's group, wanted to look at presence of tunneling nanotubes. I wanted to do 3D confocal imaging, but to simply scan an entire well at the highest magnification with 3D confocal stacks would have taken many hours just to even cover, a small amount of area. So we set up a routine where we looked for the two types of cells, determined the distance between those populations, and then limited ourselves to find cells that were within 30 microns proximity of another cell type, the other cell type, and then those locations were brought forward as the locations for high magnification confocal imaging. So the image here you can see is an example of the low magnification scan, and the analysis routine, whilst it was imaging, was able to pick out where the cell types we were looking for were in proximity, and then to show a three-dimensional render of that data, you can see the green represents one cell type, the red was the other cell type, and then shown in cyan for a little bit more clear. I would typically use blue, but you can see the presence of the tunneling nanotubes, which was just shown through a sort of membrane stain, and they were very, very happy, because it would have been exhaustive to try and find these particular events, and they were able to find enough of them to be able to draw conclusions based on what they saw. So again, just to sort of give due credit, thank you Dipali for working with me. It was actually quite good fun. So I chose to keep the talk, the actual sort of talk part that I'm giving, relatively brief. So that's the sort of some of the content of the systems I was going to go through. My contact details, fairly straightforward. It's just my name at kcl.ac.uk. We also have a website with a lot more details of the systems that we have. And to kind of go back and pause, I'll just sort of go back to the list of systems that we have. Things I didn't have time to really prepare for today, but I'll briefly talk about our multi-photon microscope. Broadly, it's a type of imaging that's very similar to confocal imaging in terms of your output, but the main difference is that you can go a lot deeper into your tissue, or in vivo imaging. So there's means there to do live imaging, or, as I say, if you really need to keep the context of your tissue and keep it relatively thick, then multiphoton is a good way to do that. We also have a light sheet microscope, which for samples up to about 2 mm cube is a relatively rapid way to scan a large volume, so that what may take many hours on a confocal or multiphoton, with the light sheet microscope you can scan larger parts. But other than just sort of talking about what we have, I open the floor to any queries, questions, or yeah.
Thank you very much, George. That was really useful to know the type of microscope systems we have in place in your imaging center. And I'm sure these are very useful information for anyone who works with microscopy. We got 2 questions on the chat. The first one from Megan. I'm just going to read it out.
Okay.
Storm image processing looks like it produces really large data files. Does that affect the development or download time?
Could you, sorry, just repeat the question again? Sorry.
Sure. So this is about storm imaging. So the storm imaging processing looks like it produces really large data files.
Right.
Does that affect the development or download time? I presume this refers to the images that has been captured.
Okay.
Yeah.
So the system we have can run the localizations while it's imaging. So you can actually get a live super resolution image as such. It's not live cell generally, although there are some live applications you can do with it. In terms of, it does produce relatively large image files. On the microscope itself, as I said, we can do the processing as it goes, or we have an offline workstation that you can do the processing on. And as part of the facility, we also have a storage solution for shuttling data around, which it has a pretty high speed connection. So typical kind of data sets where you've done a fair bit, you'd be able to upload in about 5 minutes to our storage solution, and then pull down to the workstation if you were going to do the analysis there. But I wouldn't say it's prohibitively big. It's usually the light sheet data, actually. I can't hear you, Audrey, sorry.
Sorry, I was on mute. So there's another question on the chat from Jacob. It's how easy it is to prepare, for example, stain cell samples for storm. Is it just a case of staining with specially conjugated secondary antibodies, or is there more to it?
There is more to it. At the nanoscale, you have to actually be a bit more careful about what kind of fixative you use. As it turns out, the standard fixation you only fixes about 40% of your proteins in place. And ideally you want to try and retain all of them. So there are some alternatives, such as glyoxal or a mixture of that and some other compounds. So that's one stage of one stage where you need to look at trying a few things. And then the stains that you use, it broadly varies. Alexa 647 as a fluorophore is an ideal choice to always try and use because it's very easy to get blinking. The antibodies, however, that you choose to use, the standard antibody size is about 20 nanometers, which is actually the resolution of the sort of system itself. So if you're using a primary and then a secondary, you might have your localization off target as such. So you will want to try and either conjugate to your primary, the fluorophore, where possible, with covalent bonding, or try and use a nanobody. So I believe they're developed in llamas. don't know why llamas, but llamas have tiny antibodies apparently. And they're quite small. There are other ways to tag your proteins, so you can use small N-terminal tags which will bind fluorophores. So actually your nascent protein might then just be fluorescing with the presence of the dye that you use to make them work. But there are quite a few strategies towards the sort of staining for super resolution imaging. The short summary to your answer is it's not as straightforward as normal staining, but as it's now a fairly well matured sort of imaging type, a lot of these questions have been broadly resolved, and they should now really just be aimed to be applied. It's I wouldn't go as far as to say it's very tricky, but there are things to do differently. The actual buffer that you're imaging in is often used to be the hardest thing to get right. So apart from the sort of the fixing, the staining, you also have to actually image in the correct chemical environment for the blinking to happen. And that used to be used to typically always be a system where you use a reducing agent and an enzyme which oxidizes glucose to try and reduce any presence of oxygen in the solution, and you'd make up this sort of buffer which you had to prepare fresh and put on the sample immediately to image. But there are now commercial buffer kits which make it very straightforward, and you can store your sample for up to a month to do this sort of storm imaging. So that side of things has got drastically easier because that used to be very tricky.